So the action potential that we just described in the last part of the unit was taking place in one specific region of the neuron, which we called the axon hillock, or the initial segment. But those exact same channels are located throughout the entire axon, and that same process, that same waveform is produced all throughout the entire axon. In other words, once we initiate it, the action potential has to be able to travel from the axon hillock all the way down to the axon terminal. Now, one way that we can think about this conceptually is to imagine that the entire axon is divided into tiny segments. Each segment contains all of the molecular machinery that's necessary to create the action potential right in that one local spot. Thinking about things this way, we could really describe the movement or the propagation of an action potential as spreading from segment to segment to segment through the local exchange of ions. Thinking about it this way, we can overcome the distance limitations of diffusion because the ions only have to travel a very short distance to initiate the action potential in their adjacent membrane segment. And once that membrane segment is brought to threshold, it can rely on ion exchange with the interstitial fluid. Those ions can then come in, they can diffuse a short distance into the next segment, and they can initiate the action potential there. So this is the idea that's being depicted in the image that's on the right-hand side of this slide, where three different regions of the axon are all experiencing the same changes in membrane potential, the action potential, just at slightly different times. Segment 1 goes through its action potential first, a millisecond later, segment 2 does, and a millisecond after that, segment 3 does. Now, one thing to point out here is that this is only possible because the action potential relies on voltage-gated channels, which are actually able to sense the change in voltage in their environment. This wouldn't be possible with ligand-gated channels because they're not voltage sensitive. The only thing that the ligand-gated channels care about is whether or not neurotransmitter is bound to their receptor. And that's the primary reason why the ligand-gated channels can only produce graded potentials, because neurotransmitters are only released onto specific points on the membrane, usually on the dendrites. Now that we've talked about the action potential, the steps that are in this diagram are hopefully a review. I'm going to use the numbers in this diagram as a reference point in my next couple of slides to talk about action potential propagation. So here's a representation of the axon. Again, it's being divided up into those segments like I was just talking about. And the left side represents the axon hillock. The right side represents the axon terminal. So initially, when the graded potential comes into the axon hillock, this is going to bring the voltage-gated sodium channels there to threshold. And that's going to initiate the depolarization phase of the action potential there. The sodium ions that come into that initial segment are then going to diffuse randomly in all directions, including into the adjacent membrane segment, like we're seeing here with the arrow. Now, because the membrane segment is at rest and its voltage-gated sodium channels are closed, the positively charged sodium ions from the hillock are going to serve as a depolarizing stimulus to bring that segment of the membrane to threshold. That's going to initiate the depolarization phase there. While that's happening, if you look back at the initial segment, you'll see that it's now going through its repolarization phase. And that's because the voltage-gated sodium channels there are inactivated and the voltage-gated potassium channels there are open and reaching their maximum permeability. So this process repeats as the positively charged sodium ions from the second segment rush in, diffuse in all directions, and again, moving randomly. Some of them will travel into the third segment, which is at rest. And that's going to then trigger that third segment to reach threshold and go through its depolarization phase of the action potential there. And if you look closely, you'll see that as that's happening, the second membrane segment is now in its repolarization phases. And if we go even further back to look at the axon hillock, you can see that that segment has now returned to rest. So this process repeats over and over and over again, and the action potential develops from segment to segment to segment to segment, all the way down the axon. Each individual segment fires its own action potential, but it looks identical at each point. Now, one thing that we've taken for granted up to this point is that the action potential travels in one direction, from the axon hillock down to the axon terminal. But in all the neurons that we talk about in this course, this is always going to be the case. But I just want to mention that there are exceptions. If you were to take some other neuroscience courses in our department, you would get a chance to learn about something called a back-propagating action potential, but we're not covering it in our course. We're just laying the foundations. So hopefully the animation we just went through helps to clarify why most action potentials only travel in a single direction, despite the fact that ions move randomly through Brownian motion. It wasn't until the action potential reached the third segment that the initial segment was back at rest. If we think about the states of the channels, then you'll remember that membranes at rest have voltage-gated sodium channels that are closed. Closed channels can be opened by a depolarizing stimulus. So when the initial segment is back at rest, if we were to give another depolarizing stimulus, we could actually start a second action potential that would then propagate down the axon behind the first one. But if we tried to do that in the second membrane segment, which was in the repolarization phase, we couldn't. And that's because the channels there are inactivated. Their activation gate is already open. So giving them a depolarizing stimulus wouldn't make any difference. The inactivation gate is closed, and that's not voltage sensitive at all. In other words, we could say the channels that are in the inactive state are in the refractory period. Now, keep in mind that the action potential is produced from tens of thousands of channels. So we can talk about the refractory period a little bit more specifically by saying that if all of the voltage-gated sodium channels are inactivated, then it's impossible to start a second action potential, even if we gave a depolarizing stimulus and even if we gave a strong depolarizing stimulus. This is known as the absolute refractory period. It's represented by the region shaded in pink in this diagram. If some of the channels are inactivated, but some of the channels go back to being closed, we have a mixed population, then a depolarizing stimulus would still be able to open the subset of channels that are closed. So if we had a really strong depolarizing stimulus, we could start another action potential. This is known as the relative refractory period, and its time course is indicated by the region shaded in blue. So the refractory period is ultimately what prevents most action potentials from backpropagating or traveling backwards. Even if those ions move into the previous membrane segment, the channels are most likely inactivated.